Hello, everyone. Um, my name is Jack Lasota, and this summer I've been working at the Pittsburgh Supercomputing Center. My project is on how we process, analyze, and integrate single-cell RNA sequencing data within the HuBMAP framework. This included raw data handling, open source Python-based toolkits, and approaches to align and interpret data sets across tissue types. So firstly, my project overview and background. Uh, the Pittsburgh Supercomputing Center is jointly operated by Carnegie Mellon University and the University of Pittsburgh. And at PSC, tools like Bridges 2 allow us to process large single-cell data sets quickly and reliably. From raw data to integration, my project goals and tools. The overarching goal of my project is to make large biological data sets easier to analyze and interpret. This requires that we utilize pipelines to process and output large-scale biological data, along with providing accessible tools for its interpretation. This meant standardization, or in other words, cleaning the data. So it must be uniformly processed data so that the results are comparable across both data sets and tissue types. Once we got a readable file, I used Python, particularly Jupyter Notebook, along with some open source tools such as ScanPy and Data. All the computation was run in high performance Unix environments, and I was striving for outputs that were really clean, structured data sets that were ready to be explored, visualized, or annotated. Which takes me to the HuBMAP Consortium data portal. The HuBMAP portal provides access to data sets across tissue types, even allowing comparison by various modalities such as assay type. So it lets researchers search and browse spatial and single-cell data, compare data across organs, and download data sets in raw or processed forms. So behind the scenes, creating such an interactive and user-friendly platform requires significant effort, from cleaning and standardizing the data to representing in a way that's both readable and meaningful to users. So that's where our sequencing pipelines and data interpretation comes into play. And firstly, the pipeline operation. So this graph here on the left represents a data pipeline, and each step in the diagram kind of represents a node with its own functionality where data is cyclically processed or analyzed. So this pipeline processes raw single-cell RNA sequencing data, starting from FASTQ format using the Salmon-Alvin workflow for rapid quality control, quantification, and initial analysis. The result is a clean, high-quality .h5ad file, which is very ideal for visualization and interpretation within a Jupyter notebook. So once we receive our .5h5ad files, we can start our analysis. So here I began with raw hard data, which just means it includes unfiltered, low-quality cells and genes. And I loaded into Python using data. And data just helps us track our metadata counts and analysis results together. So if we look at this picture here on the left, it shows that there's an data object with n observations times n variables. This means there are cells times genes in this data set, and there's a large amount of them, as you can see. Additionally, on the right here, I showed how the pandas library can be utilized for targeted analysis of specific data points. So I used the dot value counts function to kind of show how many cell types and gene types there are. And this is important because after filtering, we retain only high quality data. So using data in the same fashion, in the bottom left, we see that the cells times genes, about 99% of the cells have been filtered out and about 75% of the genes have been filtered. So at this point, I used ScanPy to perform clustering with the latent algorithm and was able to visualize these cell clusters using UMAPs. Furthermore, I extracted the metadata, and this is important because it can be utilized in other areas, for example, the user interface. So in the top left, we see our metadata transformed into a user-friendly portal, which allows researchers to explore tissue types, gene expression levels, and cell type specific patterns. So if we click on a particular organ, here I chose the heart, we can utilize tools like Shiny app to visualize cell and gene information. And as shown in the bottom left, the Shiny app directly uses HuBMAPs like the ones we were able to output in our process data on the last slide. So here I want to focus kind of on ScanPy itself and how powerful of a tool it is. It was the main toolkit I used for single cell analysis and really allowed me to go from raw data to interpretable clusters and annotations. And this was done in two different ways, two main ways, the ScanPy preprocessing and ScanPy integration. So preprocessing of data filters low quality cells and genes, normalizes data and visualizes gene/cell counts, identifies highly variable genes, and computes principal component analysis, or PCA, to capture major variation. Whereas data integration differs a little bit. It aligns multiple data sets in a shared feature space, projects new cells into existing PCA or HuMAP embeddings, and transfers annotations such as cell types from reference to new data. So this slide walks through the key steps of the ScanPy preprocessing pipeline applied to both heart and kidney single cell data sets. So I'll go step by step, kind of following the typical workflow used to prepare this data for analysis. So I began by filtering out low quality cells and genes. For example, I removed cells with too few total counts. Then I normalized the total counts per cell to make them comparable across the data set. And after normalization, applied a logarithmic transformation, which stabilized any variance across genes and made the data more suitable for analysis. Furthermore, I next identified the highly expressed genes. And you can see on the plot on the left the topic highly expressed genes within the total data set by the top 20. I then again used the latent algorithm to identify groups of similar cells and cluster them. So I visualized those as well with the HuMAP. And in this middle picture, you can see the clusters labeled 0 through 11. So clusters 0 through 7 here are kidney derived, while 8 through 11 were heart tissue. So using known marker genes and the highly expressed genes I identified in that first picture, I grouped them all by cell type. And using ScanPy, I created a dot plot out of all this data. And this really let me analyze mean expression of a gene within a particular cell type or cluster by their darkness. So one of those dots, the darker it is, the higher the mean expression, as well as prevalence indicated by dot size and any organ-specific differences. And this was all done before performing integration. So ScanPy really allowed uh, me to apply like the same standardized workflow across different tissues, like the heart and kidney in this example. And it produces a lot of interpretable, comparable data structures that make it easier to analyze and visualize all these biological patterns. Then finally, the data integration with ingest and BBKNN. So this slide demonstrates how we can integrate new single cell data into an existing reference using two complementary methods in ScanPy, ingest and BBKNN. So the goal is to map new single cell data, in this case peripheral blood mononuclear cells, onto a reference data set while correcting for batch effects across data sets. So what are batch effects? These are any technical differences that introduce unwanted variation in the data. So they're not any biological differences. On the left here, I show ingest mapping. And ingest mapping is a fast reference-based projection. It preserves structure and annotations. And for example, similar cell types align well, but some batch effects remain. So in this first image, you can see the CD4 and CD8 T cells, as well as the B cells, align well, but there are some batch effects, such as in the monocytes. Um, and on the right here, we see the BBKNN mapping strategy, which rebuilds the neighbor graph for deeper batch correction. So monocytes and dendritic cells kind of mix better in this example, but rare populations like the megakaryocytes could be lost. So if we look at the left one, you can kind of still see those megakaryocytes in that cluster, but on the right, it's a little bit more difficult, it kind of gets lost in the map. So these two approaches, although they serve different purposes, can really help us merge multiple data sets in a very meaningful way. And my acknowledgements, thank you to the HuBMAP Hive teams, particularly the CMU Tools component team and the PSC IEC team for their guidance and support. And a special thanks to Matt Ruffalo, Penny Kuda, and especially Zheng Li, who has helped me a lot with my presentation and just overall this summer, my sources. Thank you very much.
Yay, nicely done. I don't know if you can see all of the little claps coming up on the side, but good job. We have time for about 2 questions. If anyone has any questions, you can go ahead and unmute and ask now. Otherwise, we'll go on to our next project. That was great. All right, no questions then. What would you say was your favorite part about this, or what was your favorite thing you learned?
I think probably the coolest thing was that dot plot I was able to create because it was one of the first, I guess, outputs that I really saw everything connecting together. So that was pretty awesome.
Perfect, that's great. Again, round of applause for Jack, sorry. Congrats. All right, we'll
Thank
move
you.
on to our next presenter.